So thank you guys for coming. We're going to hear a little bit about lichens for a change, although I was pleased that they were mentioned before. Here's my general game plan as to first tell you a little bit about why lichens are important, just briefly remind you of some of those things, plus I've got an exciting new result, why they're even more important than ever. And then I'm going to give you some key findings from the first round of density management studies that we did, which was before the treatments were actually installed. And then some follow-up findings of the second round, how lichens communities have actually changed in the 13 years after thinning. So why are they important? Well, there's a lot of functional roles for lichens, and one of them is that many of them are forage lichens. Particularly long, stringy, pendulous ones are eaten by lots of different kinds of wildlife. We also have nitrogen fixers. These have cyanobacteria as the photosynthetic partner in them and contribute a lot of fixed nitrogen to ecosystems, and these can amount to about a ton dry weight per hectare in old forests. And many of the forage lichens in the previous 2 slides ago and nitrogen fixers are old growth associates. Then there's another group that you might not think of that often. These are the nitrophiles. A lot of these will be living in your backyard on the trees in town. These like nitrogen, ambient nitrogen in the environment. So we're likely to find these in polluted places in the Willamette Valley, around the edges of the Willamette Valley, and so on. And then my exciting new connection has to do with prions. You guys know about prions? Really interesting. So these are the cause of chronic wasting disease, which is an important problem for deer and elk in Wyoming, Colorado, and Wisconsin, and a few other places as well. As far as I know, it's not a problem yet in Oregon or Washington. But prions are misfolded proteins that propagate themselves. They basically cause other proteins in your brain to misfold and thereby propagate. They don't contain DNA or RNA, so they're infectious agents, but they're proteins. And these are always fatal in humans, and this Creutzfeldt-Jakob disease is one of those. And I think if you've given blood recently, I believe they ask you if you have this disease, because they don't want your blood if you do. Then the interesting thing about prions is that they're resistant to denaturation by chemical or physical agents, including things like autoclaving. You would normally think you could kill almost anything by autoclaving, but many prions are resistant to that. But it turns out, just published this year, that some lichens effectively degrade these prions. They have proteases, enzymes, that can break down the prions. And you can read about this. This was just published this spring. Degradation of disease-associated prion protein by a protease from lichens. They tested 11 different lichens, and the top 3 that they found in terms of being able to degrade these prions are shown in this picture. The bottom 2 are common lichens around here, and the one on the lower right is probably in every one of your backyards, Parmelia sulcata. So I don't know if this has anything to do with the absence of chronic wasting disease in Western Oregon or Washington, but perhaps. So some examples of things in ways in which lichens are important, and of course, like most groups of organisms, there's probably lots of ways we have no idea about yet. So on to the density management studies. So first, some key findings from the first round. You know, if you have a carload of lichen nerds and you pull up to a stand like this, people don't want to get out of the car. I mean, this is fairly boring. It's like getting home and looking in the cupboard to see what's for dinner and it's beans again. Well, this kind of forest, these high-density conifer forests in Western Oregon, Washington, definitely have a bad rap with lichen nerds. But we wanted to know, when we started these initial density management studies, if that bad rap was deserved or whether there was actually something out there that we could work with. So our initial goal was basically to evaluate these 2 scenarios. The idea was that if there were pockets of or hotspots of diversity out there, we could use creative thinning or some other stand management to enhance those pockets, for example, as leave islands, and then apply heavier treatments, heavy thins or patch cuts in the other areas. But for that to work, we basically have to find out if there are those hotspots of diversity. So our two goals for this first round were to evaluate the potential for improving diversity by creative thinning. And we also wanted then to identify structural features associated with hotspots of lichen diversity in young stands so that we could retain diversity in the hotspots while opening up the monotonous parts. And these results were published way back in 1997 by a paper by Peter Neitlich and myself. And I'm not going to go through the gory details here. I'm just going to give you some of the high-level summary here. But what we found that it's not as bad as we thought. And there actually are hotspots of diversity in fairly seemingly homogeneous forests if you go out and look for those hotspots. So then the question that keeps coming up, can we accelerate the development of old growth characteristics of these forests, in this case old growth lichens, by thinning or other management techniques? And the answer is yes. Specifically, we can avoid black holes, that is, overstocked stands with virtually no undergrowth. If we can plant in lower densities, that would help. Use pre-commercial and commercial thinning. Use variable density thinning, leaving a diversity of structural elements. And then avoid cutting trees in hotspots, including outcrops and talus areas, riparian areas, ridge tops, and sites with infertile soils because they're thin or rocky or something like that. So I want to go back to the variable density thinning and talk about what structural elements you might want to emphasize in doing that variable density thinning. These are all elements that actually have come up today in other contexts, but hardwoods, remnant trees, old shrubs, and wolf trees are all structural elements that are associated with these hotspots of lichen diversity. And I think the neat thing about this is that anybody can recognize these. You don't have to have a lichenologist on your staff to go out there and look at it and tell you where the lichens are. All you have to do is look for these structural elements and hang on to them. Put those in your leave islands, in your variable density. So far these results are really more a statement of potential rather than observing actual thinning effects, right? Because this is all pre-treatment. We're just looking at what is available, what kind of potential was there. So part 2 revisits this 13 years after those treatments were installed to see how the lichens actually changed. So here are the key findings from that part. We looked at 2 density management sites, Keel Mountain, Bottom Line. I'm not going to talk about the Keel Mountain sites because of a change in personnel associated with this, we couldn't really make the 2 sampling dates comparable. The data were not comparable enough to make comparisons. Bottom Line, we were able to do this. Bottom Line, I think many of you have been there. There's hemlock there, but it's mainly a Douglas fir dominated stand. We scored lichens on trees and branches, so just epiphytic lichens in 1 acre circular plots, and we scored them with abundance classes on pretty much a logarithmic scale. And we also measure associated characteristics of the stands in those particular one acre patches. We use several analytical tools to look at the data. One is ordination, looking at gradients in stand structure that are associated with variation in lichen community composition. Indicator species analysis, so evaluating individual species responses to thinning. And then regression models relating diversity of different functional groups of lichens to the habitat factors. And these results have been published in 2010 in a pair of papers with the lead author, my graduate student, Heather Root. The second one is on forest floor lichen and bryophyte communities. In that one, we didn't have before data, so I'm not going to talk about that. I'm just going to talk about the epiphytic where we have before and after. So this is an ordination. I imagine many of you have been exposed to these at least once, but here we go. So each of those dots up there is a plot. Each one of those is a 1-acre plot. And they're arranged according to their similarity. So two plots that are very similar to each other are close together in this diagram. Two plots that are very different from each other are far apart. The radiating arrows superimposed on that show how various other factors are related to that community similarity. And hopefully you'll be able to decipher those from the short names there. This representation explains about 64% of the variation in lichen communities on that first axis, the horizontal axis, and about 13 on the second axis, the vertical axis. And so focusing in first on this strongest axis, it's really related to gappiness. You can see that on the right, we have an abundance of nitrogen fixers or cyanobacterial lichens, which are associated with various kinds of gaps and wolf trees. And that is opposed to a pattern where we have higher conifer basal area, higher total basal area, more Douglas fir, and what we call matrix plots. So that basically the areas outside of our predetermined hotspots at the beginning of the study. The second dimension of this is more related to the presence of those remnant trees. And those are associated with forage lichens, so more old growth trees or more remnant trees, more of the forage lichens in those stands. So let's now superimpose the temporal component on this. So if you look at these solid circles in the lower left, those are the stands that were going to be thinned before they were thinned. The solid triangles are those same stands after they were thinned. So we get a general movement in response to the thinning from lower left to upper right. And then looking at the sense of those arrows that are superimposed on the diagram, you can see that that means that forage lichens are increasing, as are the nitrogen-fixing lichens, as a result of that thinning treatment. So we're getting a definite temporal change in lichen communities associated with that thinning. We can test this formally with a hypothesis test of no change in epiphytic macrolichens over time. And we're using blocked MRPP, multi-response permutation procedures. It's basically a non-parametric way of comparing communities between two groups. Null hypothesis and no difference between groups. The A statistic says that the uncut plots did differ through time. It's a fairly weak effect. That A is a measure of effect size. It's much stronger effect in the cut plots. So we detected a significant difference in lichen communities on both the cut and the uncut plots, but it was a much stronger effect on the cut plots. And on the right and lower right, indicator species analysis is showing that we're actually getting many more species increasing than decreasing in both the cut and uncut plots, but more so in the cut plots. So here are the species that really showed the most striking increases. We have several forage lichens, Bryoria, some hair-like species, a couple of the nitrophiles, which may be related to changes in air quality over that 13 years. And then cyanolichens tended to increase too, and no doubt associated with hardwoods in the stands. We only had a couple that showed decreases that they're shown here, and we don't really have a good idea of why these are decreasing in the thin plots. Then I'm just going to briefly mention the regression results. So for these different functional groups of lichens, we regressed species richness in that functional group against different habitat factors. Here's one for an example looking at cyanolichens or nitrogen-fixing lichens. They were positively associated with wolf trees at both sites, and now this is bringing back in the Keel Mountain site. They were negatively associated with hemlock and Douglas fir basal area. So in conclusion, back to the first part of the study, we could basically reject the notion that there's really no diversity out there in those young stands to work with. We do have some, and it looks like with thinning we can actually enhance the picture considerably and make a fairly rosy outcome. I would like to thank the BLM for funding the work, and Eric Shorey helped with a lot of the field work. Charlie Thompson and Peter O'Toole, Claire Hibbler, at least some of you are here, thank you, and various folks at OSU. That's it.
Hi, Bruce. Did it make any difference if a leaf island was a 1/4 acre, a 1/2 acre, or 1 acre?
We did not see that effect, but we really didn't have a big enough sample size to answer that question very well. Because, you know, most of the results, the quantitative results that I presented were just at one site only. What would have been nice would be to replicate this at more of the density management sites. We could have answered a question like that better. And actually the same thing holds true with looking at the different densities of thinning. We didn't really have a big enough sample size to address that question either. And I guess doing it at one site, you could say we only have one instance of each of those treatments.
Bruce, in terms of expanding lichens and showing up somewhere, where they come from and how far do they travel? Is there something about spatial layouts of leaf trees, leaf islands that would facilitate lichens?
Well, some lichens are very good at dispersing. Most of the old growth associates are not. Most of the old growth associates have what we would consider clunky propagules that, I mean, you're lucky if something goes, 100 yards. That's because they're reproducing by fragmentation, fairly large fragments. Other species are reproducing by ascospores, which might only be 10 microns large and actually get around quite well. Those are the species that tend to be found in the middle of something that used to be a giant clear cut. So yeah, we think of the dispersal as being a real issue with the old growth associated lichen. So yes, we think the spatial pattern is very important, and that you need some nearby propagule source for effective colonization.